In this video, we are going to be covering the topics of enzymes. Enzymes are proteins that act as biological catalysts. When it comes to the structure of an enzyme, understand that they have a small region that is called an active site, and whatever molecule is interacting with this enzyme binds at the active site to specifically catalyze the reaction that the substrate is going to undergo. So I want to specify that when it comes to biochemistry, we don't call reactants by like we call them in general chemistry and organic chemistry. So the word substrate is most commonly utilized. So remember, whenever I mention the word substrate, that's what I mean with reactant. Okay, it is the same thing. They're synonyms. It's just that in biochemistry, that's how we refer to them. So, to do a little bit of a review of what enzymes or what catalysts are, remember, as I just mentioned, enzymes are going to be these proteins that are involved in the catalysis of biological reactions. They, more importantly, catalyze nearly all, not all, but most of the chemical reactions that take place in the cell of the body. They do that by increasing the rate of the reaction because they are able to lower the activation energy of the reaction. So as we can see in the energy diagram that we have on the right side of the slide, here we are plotting the energy, meaning the potential energy for the reactants or substrates going into products. So as you can see in this reaction, we are looking at how these reactants, remember, they're called substrates. In this case, carbon dioxide and water are going to react with each other to make H2CO3, which is carbonic acid. In turn, carbonic acid is going to dissociate and we are going to be making H plus and HCO3 minus. So if we do this reaction without an enzyme, as you can see, the activation energy is being given by the top of the hill of the blue line. When the same reaction of the combination of CO2 and water making that carbonic acid, it is done with an enzyme, as you can see, the top of the hill refers to the activation energy of the same reaction. Notice that the potential energy of the reactants and products is going to be unchanged because of the use of an enzyme. The only thing that changes in the whole process is lowering the activation energy of the reaction. When it comes to enzyme names, understand that they are derived by putting ASE as the ending, okay? And usually that ASE ending that is added at times to the substrate of a particular enzyme tells you that that is the enzyme name. But that is not the only way in which we can derive names for enzymes. Sometimes understand that they can describe specifically the reaction that they catalyze. So for example, oxidases catalyzes the oxidation reaction. And there's other enzymes that simply have common names. So for example, pepsin and trypsin. These enzymes specifically are going to cut peptide bonds, okay? But sometimes enzymes even have the name of their substrate. So as you can see, sucrase, which catalyzes the hydrolysis of sucrose, has incorporated in its name the substrate that it interacts with. Enzymes can be classified based on the type of reaction that they catalyze. And there are 6 types of enzymes that you guys need to be familiar with. Number 1, oxidoreductase. So oxidoreductase enzymes are going to be involved in oxidation reduction reactions. Remember, when it comes to this, what you need to pay attention is to the reactions that we already explored in oxidation reduction. Meaning, when we have the conversion of primary, secondary alcohols to carbonyl. And again, remember that we learned that primary alcohols can be converted to aldehydes or even carboxylic acids. Secondary alcohols can be converted into ketones. That represents an oxidation reaction. So if we see those conversions, remember that gives us an example of an oxidoreductase. Also, we learn about reduction reactions, meaning the opposite. You go from your carbonyl functionality back into the alcohols. That is called reduction, which I'm symbolizing here by a capital R. We also have enzymes that are called transferases. And when it comes to transferases, they're going to be transferring groups of atoms. Most of the time when we're doing these transferases, we're transferring a group of atoms that is typically a functional group. We have hydrolases, and hydrolases are going to be involved in the process of hydrolysis. Remember that hydrolysis means that your molecules are going to be cut with water. And you know that a molecule is cut with water because the functional groups that result after hydrolysis, one is going to have an H and the other one is going to have an OH. Then we have lyases, and lyases add or remove atoms to form double bonds or to form a new ring. We also have isomerases, and then when it comes to isomerases, remember that this is going to just rearrange atoms. So whenever we have this, we're going to mostly form constitutional isomers. And remember, constitutional isomers have the same chemical formula. It's just that the attachment of the atoms is different. And then the last type of enzyme that you guys need to be familiar with is ligases. Ligases are going to be using ATP to combine two molecules. So remember, here we're going from two molecules to one large molecule. As I mentioned previously, when it comes to an enzyme, remember that enzymes have an active site. And at the active site is where the substrate binds. Remember that the substrate is what we call the reactant. Now, understand that this is a small region, and at the active site is where the catalysis of the reaction is going to be occurring. So in this example that we have on the slides, as you can see, here you have a substrate that binds the active site, and the product here goes from 1 to 2. Okay? So this is just illustrating that in this pocket, okay, where the active site, so here the green blob is sitting at the active site, we are going to have different amino acids. As you can see, I'm just going to highlight them, that are the ones that are going to be involved in the catalysis of the reaction. Whenever we have an enzyme catalyzed reaction, understand that it is very specific how we write them in biochemistry. So when it comes to enzyme catalyzed reactions, as you can see, we write them in the following manner. We have the enzyme and the substrate combine with one another to make the enzyme substrate complex. This enzyme substrate complex, as you can see, has an arrow in the reverse way because this can be reversible. The enzyme and the substrate have to come together in the correct conformation in order for this enzyme substrate complex to then go forward and produce the enzyme and the product of the reaction. Once the enzyme substrate complex is committed to make product, that reaction cannot be reversed. The reason why the enzyme is also shown in the product is because now it can go over and catalyze more reactions. It is very important to point out that in an enzyme catalyzed reaction, understand that the enzyme is just utilized in the reaction, it is never consumed or destroyed. It is always regenerated at the end, okay? Now, as you can see, the binding of a substrate occurs when it interacts with the amino acids that are going to happen, that are present, within the active site. And that's what I was illustrating here with the amino acids that were highlighted in the previous slide. Now, here we have an example of, of sucrose interacting with sucrase. First, you have the substrate interacting with the enzyme, which is this purple structure. Then we have the enzyme substrate complex coming together. Specifically, sucrase is going to hydrolyze sucrose into fructose and glucose. Then, as you can see, we have the products formed, and we regenerate a free enzyme that now it can go back and initiate catalysis of a brand new substrate. Now, there are two models for enzyme action. The first one is the lock and key model. The lock and key models work for certain enzymes. In the lock and key model, understand that your substrate and your enzyme are going to fit into one another like a lock and key. So the substrate has a shape that fits the active site shape. And because they fit with each other like a lock and key, that's how the catalysis occurs. We also have some proteins that work in an induced fit model scenario. In the induced model scenario, what happens is that the enzyme has a more flexible shape, and it wraps, meaning that it's going to change conformation itself onto the substrate. So as you can see, in the induced fit model, your enzyme is going to be more dynamic because it changes its shape to accommodate the substrate. And that's how it accomplishes the catalysis of the reaction.